Hello, I am Maxime Lafond from the Lab Tower in Lyon, France. And today I'm going to talk about ultrasonic cavitation for controlled pancreatic cancer stroma disruption. Pancreatic cancer is a very aggressive form of cancer, and the survival rates are very low, notably due to the low penetration of the drug through the tumor stroma. Ultrasound have been proposed with various mechanisms, and the mechanisms we want to develop is to use cavitation for disrupting the stroma. Notably, in this presentation, I will spend time on 3 points that are the development of an in vitro spheroid model for allowing to large parametric studies, the quantification of cavitation to allow reproducibility between systems and labs, and finally, the monitoring of the stroma disruption using shear wave elastography. So first, so this is how we do our spheroids. So basically, we are co-culturing fibroblasts with cancer cells, and we add nanoshuttles, which are nano-sized magnetic particles that are in the cells, and we incubate the cells on a magnetic field, and because they are magnetic cells now, they will aggregate in a spheroid. We can place our spheroid in the Eppendorf tube in an ultrasound machine, which comprises of 2 transducers aligned confocally, and you can also see that there is an hydrophone that will be recording cavitation noise. So here are our spheroids after 2, 4, 6, 8, and 24 hours, and we can see that they are fairly formed after 6 hours, and we can also see a good contrast that we will use in elastography. We can adjust the spheroid size by changing the initial cell numbers that are cultured. We can also see that the number of viable cells is also increasing by measuring the ATP production of the spheroids. Our cavitation quantification methods relies on calibrated hydrophones, and the way we test our method is using 2 systems that are geometrically identical, but all the measurement and amplification chain are different. We calculate a cavitation index that will be homogeneous to kilopascals based on the average pressure in the measured in specific frequency beams. And then we will compare the relations between the driving voltage, cavitation index, and regeneration of radical oxygen species measured by fluorescence. So here on the upper left is the cavitation index, homogeneous to kilopascals as a function of the voltage, driving voltage to the transducers, and this is for 2 systems. So we can see that those systems are really different. They are producing cavitation noise that's really different from one system to the other. If now we look at the cavitation activity by looking at the fluorescence of the radical oxygen species solution, you can also see that the cavitation activity produced by as a function of the driving voltage is different. So we cannot rely just on the driving voltage when going from one system to the other. But what we can see is that with our method of calibration, now the fluorescence as a function of the cavitation index in kilopascals, the 2 systems, so the system 1 with the dots and system 2 with the cross, you have all this region where they intersect. You can see that we have outliers, which is probably due to the plateauing of the cavitation noise with increasing driving voltage. So it's not sending more acoustic power, but it's doing more cavitation, as we can see on the fluorescence data. So it's just the limit of that system that when we reach that plateau. Finally, the elastography to measure that we place our spheroid in a plate under 10x magnification microscope that's plugged to an ultra-fast camera. And we send a magnetic impulse to make the spheroid oscillate because it's charged with magnetic particles, and we can so take those images, and from those images check the displacement and the shear waves in the video. Here are two representative images. Well, that's actually just one spheroid, but before and after ultrasound. So we can see that the speed of the shear waves is decreasing. All those white areas are because there was not enough signal in those areas. And we can see, we can compare before and after placing spheroids in the system in the control group. So without ultrasound, there is no change. But in the group that receives cavitation, we can see a sharp decrease of the velocity of the shear waves, which means they are now softer. To sum up, spheroid allows the assessment of a large number of parameters. We designed a quantitative cavitation regulation that should increase reproducibility between experiments and systems and potentially labs. And finally, we showed stroma disruption in spheroid. The next steps will be to now evaluate the combination of gemcitabine and cavitation with viability assays and monitor the elasticity modulation in vivo on a orthotopic pancreatic tumor model that we've developed with our partners. Thank you for your attention, and thank you to our sponsors.